Appreciate it. Hi, everyone. First of all, thank you for inviting. It's always great to come and share our data, whatever we've been doing for the past year or more than a year. What I'd like to do today is basically walk through 3 different chapters of the work that we do in the lab. And all of it is kind of focused around optical mapping and structural variant identification on the genetic level and some epigenetic level for copy number variant identification. A disclaimer that everything I show today has nothing to do with clinical application. It's only research for now. So again, first part, I want to quickly go over some of the structural variants that optical mapping is really good at identifying, and some of the validation that we have done with case studies of Duchenne muscular dystrophy. Second part is evaluating structural variants in specific genetic disorders of sex development, and undiagnosed diseases. And then part 3, investigate haplotype-specific epigenetic changes using optical mapping as a basis of identifying long DNA molecules and epigenetic methylation levels that come with it. So we're all familiar with the human genome, 23 pairs of chromosomes, more than 6,000 different genetic disorders identified to date. Most of we focusing on Mendelian inheritance, so one gene, one phenotype, or multiple phenotypes that come with it. These affect millions of people around the world, and with the low prices for short-read sequencing, what we're seeing is that millions of people in the next 10 years are going to go through these Illumina sequencing to get their genetic diagnosis. So the reason behind it is basically the introduction of precision health, which aims to get your phenotypic information, your family history, your habits at home, whatever you eat and drink, combine that with your genome, find which susceptibility genes you have, and prescribe basically the pills that you need to get better or stay healthy. But one of the main limitations of this approach, which is the short-read sequencing paired and 150 base pair reads, is that the human genome is quite repetitive and it is missing on identification of large structural variants. Even in non-repetitive regions, whenever you have unique sequences, the algorithms that are currently used are not very good at picking up structural variants. So if we look at the construction of the genome, we see that 1/3 of it is unique sequences, and the largest part, 2/3, is made up of highly repetitive elements in different sizes, from 1 kb to all the way to tens of kilobase pairs. So it's important to understand that when we use 150 base pairs of paired end sequencing, anything that is aligning here is not really good for identifying any structural variants or any type of variants. And then this part is actually what's mainly used for whole genome sequencing. So when we work with exome sequencing for my PhD work and continuously afterwards, and the work that we do in the lab now, we continuously use exome sequencing and genome sequencing, and they mostly are missing those structural variants that are in repetitive regions. So if we look at these 2 technologies are primarily used in clinic and around the world, we see that we can, for array-based applications, we can get anything that is gain of material, loss of material that is greater than usually in clinical realm. Anything that is greater than 30 kilobase pairs is usually reported to a patient. And then for Illumina sequencing, it's really good at identifying single nucleotide variants in common region, in unique sequences, but not in the repetitive sequences. But when it comes to structural variant identification, it's not really good. So when we were deciding to move from Illumina, but use Illumina in addition to some other technology to identify different types of variations, we were looking at long-read sequencers, for example, the PacBio and Oxford Nanopore, and as well as optical mapping. We were wondering to identify the best suited technology to not only identify single nucleotide variants and small indels, but also find structural variants of different sizes. So here, what I basically highlight here on the bottom is more or less the average cost for a single human genome for long-read sequences using PacBio or Oxford Nanopore, and then here is optical mapping. And what I want to highlight is here the read lengths that are actually most important here. So with the Pacific Biosciences and Oxford Nanopore, you more or less get the same amount of read lengths. So if you were to generate, let's say, 90 gigabase pair run, you would get your average read lengths would be 5 to 50 base pairs for Pacific Biosciences and 10 to 50 kilobase pairs for Oxford Nanopore. Now, when it comes to repetitive regions, this is not terrible because it's a lot better than short-read sequencing. But again, when you go to line elements and really repetitive regions, it's not enough to cover those. So and the prices for generating this data is a lot more expensive than optical mapping. So what we hope to accomplish for the next cases that we analyze is incorporate short-read sequencing for high accuracy of single nucleotide variant identification with optical mapping and expression data to be able to find small variants, large variants, and then use the expression values in order to link them to any variants that are outside of genes. So here's a representation of one of the slides that Sven showed earlier. Here, what I want you to focus on is 3 different colors: the pinks, the greens, and the blacks. So here, this circle is basically showing deletions on the left side, insertions on the right side, and we can see that short-read sequencing, even though it's able to pick up some of the variants throughout the different size lengths, it's really missing the bulk of it in both directions. More on the insertion side than deletion, but a lot of the variation is still missed. When we look at anything that is greater than 500 base pairs, we see that the Bionano is picking up a lot of these large structural variants, which in the human genome, if we take the gross number of base pairs once, is the largest difference between human to human variation. So it is important to be able to capture these when you're trying to find causative variants. So before starting, before using optical mapping, it was important for us, for our own sake, to identify to validate the method and to understand that it can actually find structural variants for our needs. And for that, we used a cohort of cases with Duchenne muscular dystrophy, which is a form of muscle degeneration, usually X-linked and affects males around the age of 3 and 4. And so here is a representation of reference map. Again, everybody's familiar with this, but just quickly go over it. The reference HG19 is in blue, and then the sample maps are in yellow here. And we can see that we can identify precisely which exons have been deleted for the Duchenne gene. And we can do that for basically almost any type of size that is greater than 1 kb. Here's an interesting case of almost 800 kb duplication that we have identified that is in tandem within the DMD gene that kind of disrupts the protein function. So here, one thing I'll mention on this slide is because we've spent, actually, after we identified the size of this, we've spent about a month and a half trying to use long-range PCRs to validate the breakpoints. And so that kind of comes back to what Amir was explaining, that why there's a need to incorporate both of the technologies together. So for this particular case, we did have the genome sequencing, and when we run it, we do see that there is a gain of material, which is not the correct sizing here. But when we run the integration pipeline, we narrow down the breakpoint resolution so that it's easier for us to validate with PCR and get back to the patient in the clinic. The second example here, the third example, is inversion. Again, this is 5.1 megabase inversion that it disrupts the DMD gene. So kind of proves the method that you can, if you align your map with the reference map, you're able to find any irregularities between them. Again, in the clinical environment, this took a while to diagnose, and with optical mapping, it's about less than a week. And then the last example here is quite interesting. This one, we identified a 420 kilobase pair insertion within the DMD gene. And upon closer examination, this is this part right here between exon 44 and 45, we identified that this region is actually inversely mapping to chromosome 15 in an inverted orientation. We identified that there is enough copies, enough material or molecules to represent that this region actually has been duplicated from chromosome 15 and inserted in inverted orientation on chromosome X, where the Duchenne gene is, and disrupted the formation of the dystrophin. If we were to run this on the microarray, we would have picked up the gain of material, but we would not know where in the genome it has been inserted. And we did have whole genome for this, and we've run it, and again, it was picked up wrong sizing of the duplication, and we did not find that it was inserted on the chromosome X. So it's important to have this technology in order to be able to say, yes, I do have a gain of material, but where does in the genome it go? Okay, so now transitioning from the validation to genetic diagnosis, we basically wanted to evaluate the method when you go genome-wide, and you don't know where you want to look. How do you take these structural variants and analyze them? So for that, I'm going to focus mainly on disorder of sex development and undiagnosed diseases here. So if we look at the ASHG 19, I think this is the reference here, we see that a lot of the molecules, this is a typical run of ADX coverage and molecule lengths here, we see that we have a really good coverage for the entire genome from chromosome 1 to 22 and X and Ys, except the centromeric regions and the long arm of Y chromosome, which is the sequences are not well known. So if there is, if we have molecules and the pattern of the enzyme that it was used to align, we would not be able to align because there is no sequence. But it doesn't mean that the enzyme that we're using doesn't recognize those sequences, and we don't have any molecules there. So coming back to Amir's presentation, here's like a little, my way of representing it is basically once we get the optical mapping and we have whole genome sequencing, what we typically do is we'll get the breakpoints from OGM, the M kind of shifted here, and then we'll get the SV calling done using the self pipeline from Bionano. These breakpoints are going to be used to realign some of the short reads in the breakpoint area using the AI that Amir talked about, and then that will be going back. Both of these are going to be used for joint SV calling, and so that kind of helps to identify, to at least increase the breakpoint resolution for unique sequences wherever we find structural variants. And currently the work has been done for these three categories, and then inversions should be coming quite soon. One way I look at this integration as a beneficial pipeline is that it decreases the breakpoint uncertainty from kilobase pairs to base pairs, and I'm not going to talk about the numbers because we don't have them for now. But what this helps us to do is, like, for example, if we find a deletion upstream of a gene and we have a 3 kilobase pair window, whether it lands on the left or right of the promoter region or the protein or gene strut, then it's important to know exactly where the base pair is to identify if it's pathogenic or if it affects the gene or not. And then the second is that to validate some variants that are lower than 500 base pairs. So Bionano, if you look at the structural variants that Bionano identifies, routinely you'll find variants that are below 500 base pairs. So to us, it was important to understand, if we have the short read sequencing data, can we leverage some of these smaller base pair structural variants to try to use short reads to validate some of these and increase the sensitivity. When we look at the number of variants identified, I apologize because I haven't updated these numbers. There might be more of these whenever you run the DLE solve pipeline these days. So the number, as you can, I'm not going to read them through, but there's a lot of variants that you identify, and in order for you to sift through them, you need to have some sort of annotation pipeline reference, frequency information for the population-based analysis. And if we plot them through the insertions and deletions, what we see is typically the number of insertions is much higher than number of deletions, and this is kind of indicative of the reference genome incompleteness. So for example, if the genome was kind of representative of the population that we're studying, we should have been able to basically kind of have similar numbers of insertions and deletions. So once we have these numbers, what basically we've developed, Surajit Bhattacharya is here on the corner, so if you want to talk to him about more how this works, we developed this R package that is called Nanotator, which is a structural variant annotation, where it takes basically the SMAP input that has the refined breakpoints from Genox, and then basically provides which information, basically the information here. So if there is a promoter deletion, for example, the line here, we'll see which gene is right next to it, provide population frequency, cohort frequency, this is the internal frequency, the number of samples that we've used. If there's any gene expression information that we have for that sample from blood or from fibroblast or whatever tissue of interest we have, that information is going to be incorporated for that gene X. So for example, if the proband had 2 TPMs, that gene X will be printed here as 2, and then RNA-seq for the father and mother are going to be right next to it. In addition to that, if there is any phenotype associated with that gene, we're also going to print it in the phenotype area. And then the second example is very similar. There's a structural variant that occurs in the gene. We print all this information, and that kind of helps us to classify variants and prioritize. So here I just want to show, a lot of people generally ask how is structural variant analysis different from short read sequencing analysis for small variants. And in reality, it's exactly the same thing. So we start with DNAs. We use a different kit, not a column-based kit, to extract high molecular weight DNA. We label DNA, push it through the Sapphire instrument, and then we get a BNX file, which is kind of equivalent of a FASTQ. The FASTQ is then used in the Access server that Alex showed in the morning for QC, and then we push that through the compute server for genome assembly and variant calling, and then you choose your reference of interest, and then you get the SMAP, which is the structural variant or the VCF equivalent. The VCF, then we use the genox integration pipeline. If we have parents as well, we'll incorporate that. Oh, I'm sorry. Before going to the genox, we'll basically run the variant annotation pipeline that Andy was working on. So what this helps us is to take the structural variants that are occurring in the proband, and we'll understand whether these are present in the mom or dad. And I really like the new update that you guys have, the Bionano has, where it tells you how many molecules are spanning that in the mom and dad. It really is helpful to understand whether it's a de novo variant or inherited. And then we use the short read integration with genox, and then we do annotation with annotator that kind of generates our last Excel that has different sheets in it that has de novo mutations, homozygous mutations, compound heterozygous mutations, and the variants that are inherited either from mom or dad, and then any genes that were in our gene list of interest. So now I'll basically go through some DSD examples, some undiagnosed examples, and show what we've found so far. So this case presented in clinic as a possible Klinefelter syndrome, which is a 47XXY case. These is usually, the phenotypic appearance is usually a male looking phenotype. These individuals usually have learning difficulties and intellectual disability with some variation. Low muscle tone, female-like fat distribution, hypogonadism. Mostly, some individuals have infertility, but it's still possible to have children with IVF. And some defects may affect the heart and bones. So we've extracted high molecular weight DNA. We've run it through the regular DLE pipeline, and what we found is that this particular case, instead of being a Klinefelter syndrome, had actually 4 X chromosomes, and that's kind of represented here, the X chromosome, and then the number of chromosomes, the copies here. So you can see this one has 4 X chromosomes and 1 Y here, and then these are just the regular control wild-type male and wild-type females here. So this is actually a very well-known case. So it's a little more severe form of the Klinefelter, but the phenotypic presentation is quite similar. The second case was 46XX Wager syndrome, which is a Wilms tumor. The Wilms tumor is usually a kidney disorder where you have a tumor on your kidneys, but it's also Wilms tumor 1 gene is usually also associated with ambiguous genitalia, and these Wager syndrome usually comes with aniridia, which is the absence of iris in your eyes, very typical for the syndrome, genitourinary abnormalities, and then intellectual disability. Initially labeled as mental retardation, that's why the R, not I. So here we've identified a 6 megabase deletion in the locus of Wager syndrome here, which included the WT1 and the PAX6 gene that are typically present in Wager syndrome deletions, and this is autosomal dominant, so you just have one of the copies that has to have this deletion in order to be affected. And since our case was not a male, we did not see any external genitalia abnormalities, so the external genitalia looked like a female. But in females, 46XX cases, many times the internal gonads are streaked, so they're not producing any eggs. And then the third and fourth case are actually kind of investigation cases where we have not reached the diagnosis, but we have a gene that is kind of interesting, and it could be a new gene discovery. So this case is a syndromic form of 46XY gonadal dysgenesis. Externally looking like a female, no palpable testicles and no uterus, has some bone issues and growth delay, elevated gonadotrophins, and negative clinical exome analysis. So we've run it through the regular pipeline. This one, I don't remember whether it was DLE or endonuclease based, but we identified a 6 kilobase pair insertion in WDR11 gene, which has previously been associated with hypogonadotrophic hypogonadism in autosomal dominant pattern. The only thing that and this person is actually 46XY, we did not have the father for this, so that we don't know whether the father is carrier for this, so it's a little difficult for us to call this as a de novo variant and diagnose it. So it's a research And the phenotype is actually fitting somewhat well, but we don't know exactly where it lies. So we'll have to do either long-range PCR to find the insertion or incorporate it with whole genome. The last case of DSD that I'm showing here is a 46XX female with ovotestes, which is basically some masses in the gonads. You have testicular tissue as well as ovarian tissue, and we identified a 165 kilobase pair insertion here that it seems to include, so disregard this this green. It's upstream of PDGFA, but because there are no labels in that gain of material region, we don't really know if it's actually duplication of PDGFA, but we do know that the receptor of this PDGFA gene has been associated with Dennis-Drash syndrome, and homozygous deletions of its receptor gene in mice results in peritestes. So one of the things that we're thinking that if it's a duplication of this gene, that it could be that it actually drives some of the ovotesticular formation. But again, a research variant. So the UDN cases, his undiagnosed case of first case is a 5-year-old male with infantile spasms, severe hypotonia, long eyelashes, prominent jaw, generalized epilepsy, and neurodevelopmental delay, with MRI showing some brain malformations. When we first, typically when we run the optical mapping, we'll take a look at the exon 1, make sure that the karyotype sticks around. So here what we identified on the X chromosome, first time I looked at it was quite interesting, where we looked 2 maps. Generally, if you're XY, you're going to have just one long map representing the X chromosome. But what we saw was 2 contacts aligning to this region, one of them showing 89 kilobase pair deletion, and then the other one aligning to the reference just fine. And when we look at the molecules, there seems to be enough support for both of these maps. So contacted Bionano support, and what we came to a conclusion that this is actually a mosaic variant that takes out first half of CDKL5 gene, which has a lot of overlap with the patient's phenotype. So this is a kind of interesting, I would say, autosomal dominant way of diagnosing this case. So if we look at the gene, this is this CDKL5 gene is most active in the brain, is essential for normal brain development. So there is, if you look at the OMIM, you'll find all of these things that are kind of present in our case as well. So it's right now we just need a way to validate the variant, and that should be a good diagnosis. The second case is a 13-year-old female with hypokalemia, hypothermia, failure to thrive, G-tube inside, scoliosis. I don't know what that is. Anyway, insomnia, idiopathic fatigue, and so again, we had whole genome for this case. That was negative. We have run optical mapping, and what we identified is a de novo mutation in this gene that is 14 kilobase pairs in size. But we also identified 4 extra copies of 100 kb repeats right upstream of it here in this region, and I haven't added the colors, but basically this region is mapping to this place 4 times, here, here, and here, and here. And so we've contacted the treating physicians, and they said that the hypokalemia with this gene is kind of a known thing because of the recent publications that show that mutations in these genes are associated with hypokalemia. And so this is an interesting variant that is currently researched. We need to find out how exactly these extra 4 copies and this copy here influences the phenotype in the patient. And then the last case is for the UDN is quite fascinating. Actually, this case has a lot of different abnormalities. I'm not going to read through them, but there's basically a syndromic case of a lot of different things that are happening in the case. And so once we run the structural variant calling, we identified that in the BMPAR gene there is a 10 kilobase pair deletion in one of the alleles that is coming from the mother because we have the parents. And we identified that this gene is likely taking out a couple of exons from this case. And we also identified that that this sample from the genome has exon 2, just outside of exon 2, has a heterozygous variant point mutation that has a 0 frequency in our in the reference databases from Nomad, Exact, and TopMed. Which was classified by GenOx and ACMG guidelines as the VUS. So So when we look at the omen, we see that this gene actually has a lot of, I'm assuming no one can read this, right? Okay, never mind then. All right, so there's a lot of overlap with the phenotype that this gene has in autosomal recessive pattern with the patient's phenotype. So, we need to validate the variant with the Sanger sequencing and see if we can have the diagnosis for this case with this gene. And then the last one is I'm going to show, this is a compounded heterozygous variant in the Gittleman syndrome, which is also a kidney disorder that causes imbalance of charge atoms. Here, what we see is we didn't have the parents for this case, so we don't know which one is coming from which. We identified 23 kilobase pair deletion in one of the alleles, and then 27 kilobase pair duplication in the other allele. So the reason you're seeing two maps here for each of the alleles is because this one was run using endonucleases, so we generally use two endonucleases for this. All right, so that's part two. And then the last part what I'm going to talk about is how to leverage the length of the molecules in repetitive regions to understand the epigenetic profile of some of the diseases we're studying. Here, this method was originally developed by Yuval Ebenstein from Israel and in collaboration with Bionano, where basically the method is based on endonuclease labeling of long molecules, and then the newer method of methyltransferase-based DLE labeling. So first step, you will basically use either BSPQI or BSSI enzyme that is specific to your genome, and you can use any other nucleases that, if you're using non-human, that is better, has better label density, and we will label them with red labels instead of using green labels, repair this DNA, and then the second level methylation-specific labeling will be done using MTAC1 methyltransferase that recognizes this TCGA sequences, and whenever there is a methylation on the C, then no green label is added, and whenever there's the C is not methylated, then we add a green label. So we have, at the end, we have two levels. One is for the genomic level for genome assembly, and then the second one is green for the methylation quantification, and then the DNA back one is labeled with blue stain just as a regular procedure. So the way this, again, showing kind of like exactly how it works, if this TCGA sequence in the C, there is methylation non-present on the C, then this fluorophore, which is the green color, is being added to the adenine. And if it's methylated, then this reaction doesn't proceed. So it's an indirect way of measuring methylation where you actually measure hypomethylation rather than methylation. So anytime you see a green label, it's actually showing that the region is hypomethylated rather than methylated. So the running it through the nanochannels is basically exactly the same after you've prepared it. Here is an example of how the molecules look. You have a DNA backbone in red, and then two colors green representing the methylation-specific labeling, and then the red for the backbone and enzyme, which could be either BSPQ or BSS, the one that you used. And so the assembly is primarily done using the red channels here, and then the green channels are condensated and basically put inside the nicking points to quantify the non-methylation. So here again, we brought an R package. I don't think it's available yet, but it should be available soon, which basically takes the information from the contig information that Bionano is assembling, and it goes back to the molecules, and it finds where the nicking positions were aligned to the contigs, and it quantifies how much green labels are between them. And then at the end, it spits out this graph that kind of says between this label and this label, we have that many green labels, and kind of quantifies how much non-methylation you're having in that region. There are two main advantages to this dual labeling in the context of disease. One is quantification of methylation in repetitive regions, and I'll show you an example of this for FSHD. And then we can study the effects of methylation in repetitive elements, which is quite interesting. And then the second one is haplotype-specific methylation patterns, where we can study the imprinting from maternal to paternal allele. So here, I know a lot of people have talked about the FSHD today. Did anyone? I don't know if anyone mentioned, but it's a muscle degeneration that usually starts around the age of 20 and affects males and females in about the same way. Facial muscles, scapular muscles here, And it has a prevalence of 1 in 7,000, and the main driver of this is hypomethylation of the D4Z4 repeat array on the 4q A allele. And I'll show you basically, this right here is a BSSI label of the D4Z4 allele without the secondary epigenetic level. And so what I want to show here is that we are cleanly detecting the repeat expansion, which is 3.3 kilobase pairs inside on one of the alleles, and we're detecting the deletion on the second allele. And so what we were interested to find out in this configuration is what do the methylation levels look in each of the repeats, as well as the beginning of the DOX4 gene, which when expressed is causing the disorder. And then the secondary reason for having the methylation in these cases is to understand so that the 95% of the cases are caused by the constriction of this region, but there's 5% of the cases that have mutation in SMCHD1 gene, which causes hypomethylation in this region without the needing to have the constriction. So if we access the epigenetic level information as well as the constriction, then basically we can pretty much diagnose almost all cases. So what we did as a trial run, we ran siblings, one case, this case number 1, that was affected with this disease. We found that this case had 4 repeats in the area, and 4qa allele, which is the permissive allele, and then the second allele did not have that, and we plotted basically the amount of hypomethylation that we see in here. So the red peaks represent the short allele, the first 4 repeats, and the DOX4 region, and then the green repeats are for the longer allele and how much hypomethylation we see in all of those. So what we've noticed that for the short allele, we have the levels of hypomethylation are typically about 2-fold higher than the normal allele. And so when we looked at the unaffected sibling, this was not kind of replicated because both alleles are normal and there's no constriction. So we decided to run more samples and average the methylation levels in order to understand if in fact the method can be used for identifying, basically diagnosing the FSHD using only the methylation patterns. So here in this image what you see is an average, this is N of 3, so basically we have 3 4qa alleles that have the constriction, which is less than 10 repeats. And then we are plotting this against the same sample that has the longer alleles. And what we see is that this D0, which is the gene start and includes the gene body of DOX4, we see about 2 times higher hypomethylation levels, basically number of labels. But we didn't replicate the first finding that we saw, that the actual repeat themselves also had more hypomethylation than longer alleles. When we plot this in more easier to understand way, what we see is that if you have an affected allele, you do have more labels in there when you normalize them against your number of molecules that are spanning that region. But if you have higher than 10, then the methylation levels are lower. Non-methylation levels are normal. This is not quite significant yet, but I would say that this is N of 3 only. And if we remove one sample, it does become significant. And that one sample that I'm talking about had a lower than expected coverage, so it might still be significant. And then the last case I'll talk about is Beckwith-Whitman syndrome for the imprinting of paternal allele that we have. Beckwith-Whitman syndrome is an overgrowth syndrome that affects many parts of the body. Some parts may grow abnormally in terms of the size of the organs, or you might have a larger tongue, and increased risk of developing cancer. So if you don't know how this basically works, is you have a maternal allele and a paternal allele, which have 2 regions that have imprinted methylation sites, IC2 and IC1, and in 50% of the cases, the methylation in this IC2 is lost, which drives the expression of this KSTNQ1, and that results in this Beckwith-Whitman overgrowth syndrome. I forgot the percent, but in other cases, I see one gain of methylation from the paternal allele also results in this. And what we wanted to know in this particular case, there was an indication that the paternal allele was fine, but it had a duplication of this region that resulted in having 2 copies of expression of this gene. So here what we have identified between IGF2 and HS19 that the maternal allele was normal, and then the paternal allele had duplication of this region from here to here that presumably carries the methylation states of the paternal allele. So if we look at the molecules, again, I know this has not been quantified at all. So what we're expecting to see that the methylation levels from the maternal allele will have a different pattern, and then whatever methylation patterns we have in this duplicated region will be consistent to the paternal allele. And that's it. So I'd like to thank the laboratory, all of our laboratory members, especially Surajit, who wrote some of the packages needed to analyze this case. Want to thank Bionano for all the support, those weekends calls and bugging you for answers, and Genox for integration pipeline, really working well. Our collaborators at UDN, at UCLA, and Duke University, and as well disorder of sex development. So, and I'll take questions.